Dear students, welcome to CSIR Virtual Laboratory Video Demonstration on Isolation, Culture and Functional Characterization of Stem Cells. This video is presented by CGCRI, Kolkata. We know stem cells are undifferentiated cells with the capacity to both differentiate and multiply into 200 cell types that form a human being. We also know stem cells have various clinical applications like defects, missing teeth, bone marrow transplantation, spinal cord injury, blindness, stiffness, muscular dystrophy, and diabetes. And to make it clinically suitable for different purposes, we need to first isolate them from its cell mass. Isolation of stem cells. We can isolate stem cells mainly from two sources, that is, adult body tissues and embryos. Some other sources are cord blood stem cells and amniotic fluid stem cells. Scientists are working to develop stem cells from other cells using genetic reprogramming technique. Since embryonic stem cells have great therapeutic potential and can be obtained from the embryos that are 3 to 5 days old, so its isolation from inner cell mass is a big challenge. Researchers reported that ICM can be obtained either from fresh or frozen embryos. Thereafter, several methods are developed for this isolation, which include mechanical dissection, where ICM is isolated by mechanical pressure. ICM can also be isolated by laser dissection and by using immunosurgery method. Embryonic stem cell can also be obtained from ICM by microdissection of blastocyst using fine needles. Laser-assisted biopsy is also one of the most promising techniques for isolation of ICM. Unlike ESC, adult stem cells are stored within our body and regenerated heavily when they get signal culture. After inner cell mass isolation, the stem cells are grown to generate embryonic stem cell using feeder layer, extracellular matrix, protein, peptides, and synthetic polymers. Embryonic stem cells were first cultured on a feeder layer of embryonic fibroblast in medium containing serum. But nowadays, most stem cell culture use defined serum, free media containing various additive and growth factors. Stem cells are then frozen by a special program and preserved in liquid nitrogen at -196 degrees Celsius. This method of preservation of stem cells is called cryopreservation. Functional characterization of stem cells. The preponderance of current somatic cell research focuses on the characterization and identification of stem and progenitor cells in various tissues. However, stem cells can really only be defined by their functional role in their tissues of origin, that is, their ability to give rise to all other cells in the tissues. To achieve this, stem cells must actively traverse the cell cycle and undergo both symmetric and asymmetric cell divisions. Hypothetically, the types of stem cell based on their functions are totipotent. These stem cells can differentiate into all possible cell types. The first few cells that appear as a zygote starts to divide are totipotent. Pluripotent. These cells can turn into almost any cell. Cells from the early embryo are pluripotent. Multipotent. These cells can differentiate into a closely related family of cells. Adult hematopoietic stem cells, for example, can become red and white blood cells or platelets. Oligopotent. These can differentiate into few different cell types. Adult lymphoid or myeloid stem cells can do this. Unipotent. These can also produce cells of one kind, which is their own type. However, they are stem cells because they can renew themselves. Examples include adult muscle stem cells. Nowadays, scientists see many possible uses for stem cells like tissue regeneration, preparation of different drugs, toxicology screening, blood disease treatment, cell deficiency therapy, and brain disease treatment. Thank you.